about communication, self-communication. It's going to be a long video, so if you are one of my students, you should have your notes in front of you and you're taking notes as you are watching and listening to the video. And it's going to be a long one. I haven't recorded a video in a long time, so I'm a little nervous because I don't remember how the whole screen recording thing works. Hopefully I won't be talking to myself for 3 hours. Oh gosh, 3 hours and then it not record anything. That would be a nightmare. But I think it's recording. All right, let me go ahead and minimize myself so we can take a look at the PowerPoint. All right, and play from current slide. Okay, I think it looks good. I hope so, fingers crossed. So we are going to talk about communication, cell communication. So what is cell communication? It is a process by which cells detect and respond to signals from the environment or other cells, okay? So, for example, it's happening right now where your sensory neurons, like your auditory sensory neurons in your ears, or the visual sensory neurons in your eyes, you're detecting information coming in and it's being processed. Those signals coming in from the environment, the information is being processed and communicated to the rest of your brain, okay? And you're processing that information. Another example of cell communication is with bacteria. You know, bacteria, their lifespan is very short, so they have to be able to utilize whatever food is available right then and right now. And so they can detect from their environment nutrient availability, and they can make any metabolic adjustments. What I mean by that is we talked about in class how bacteria, they actually prefer to digest or metabolize, I should say, metabolize glucose over lactose. And so what they will do is they will adjust transcription of the lac operon in response to whether or not glucose or lactose is available. And then also, the cells within your body, your multicellular body, they're communicating right now to coordinate the activities of everything that's going on right now in your body. Okay? All right. So a classic example of communication that's happening in your body is the fight or flight response. So this response is activated when your life is in danger. So just imagine a scary moment, and hopefully you haven't experienced this too many times, but a scary moment where you feel like your life is in danger, the amygdala, which is part of your brain, reacts to the threat, and then it sends a message to the hypothalamus. And the hypothalamus is going to activate what we call the sympathetic nervous system, the autonomic because you can't control it, and it sends a message all the way down to your adrenal glands. And the adrenal gland, and let me just get my marker here, the adrenal gland sits right over here on top of your kidneys. Okay, so you have two of them because you have two kidneys and you have two adrenal glands. The adrenal glands, they release epinephrine or adrenaline. Epinephrine, adrenaline is the same thing. And adrenaline, epinephrine, those are hormones that target all over your body. They target like your heart cell, they go up through your bloodstream, they target your eyes, okay, and specifically in your eyes, they target the pupils, okay, the muscles that can open and close your pupils, and your pupils will actually dilate. Dilate means to open up, and so you'll temporarily have better, improved eyesight when your life is in danger. And that kind of makes sense, right? If your life's in danger, you want to be able to see more, get away from the danger or fight back. Okay, and then it also sends a message to the epinephrine. It targets your salivary glands, and it causes the salivary glands to stop releasing saliva, and you end up with a dry mouth, right? Whenever you're nervous, don't you get a dry mouth? Like when you have to do a presentation in front of your fellow peers in the class. And then also it targets your breathing. Okay, your breathing will increase. Really fast breathing, and then your heart is pounding. It targets your heart, it targets your muscles, they get all tense. And then if you have food in your stomach, You might feel sick to your stomach. I'm sure you guys have heard that before. So adrenaline actually causes digestion to slow down. And if there's food in your stomach, it makes you feel a little bit sick because the digestion is no longer happening. Okay. And then also it targets sweat glands to start sweating, and you get all like nervous and sweaty palms. Okay. And this is all part of the fight or flight response. Next, maybe the fight or flight response applies to when you're taking a test, an AP bio exam. It sends a message to, the adrenal gland to release the epinephrine, and you get all sweaty and nervous, etc. Heart pounding. You can relate, right? So how does communication happen on a cellular level? Okay, there's basically 3 parts to cell communication, and we also say. We also describe this diagram about how a signal message gets into a cell. We call this a signal transduction pathway as well. So signal transduction pathway, and I apologize, my writing is so messy. But what's happening is that the signal molecule, or a ligand, okay, that's something that you guys need to know. It's a vocabulary term, ligand. Ligand is a signal molecule. It binds to a receptor protein. See, this is a protein, a transmembrane protein that spans a cell membrane, the phospholipid bilayer, the plasma membrane. And what happens is when the ligand binds to the receptor protein, it causes the receptor protein to change shape. We say conformational change. It changes conformational shape. When it changes shape, it transmits a signal into the cytoplasm, because this part of the protein, it changes shape. And then because it changes shape, there's something that happens in the cytoplasm activating other molecules and enzymes. And when it does this, we call that transduction. The message gets into the cytoplasm. We call that transduction. And these molecules, and you can see in this pathway, there's several of them involved activating each other. It's sort of like we call it a cascade. We call it like a, I'm running a blank here, but it's like a cascade, okay? And then the response part, so that's the last step, the cell responds. So somehow this signal that's outside the cell, and it stays outside the cell, okay? This signal molecule, it stays outside the cell, it is received by the receptor protein, and it's going to eventually cause the cell to respond in a specific way. And for example, the epinephrine in the fight or flight response, it targets the pupillary like muscle cells, and it causes the muscle to contract, okay, or dilate. Actually, it causes it to dilate, okay. Let's see. In cellular systems, signals are generally chemical molecules like hormones, like I mentioned, epinephrine, but it could also be light, okay, an environmental condition like light. Pathways involved in communication are called signal transduction pathways. So in this case, the signal is light, and when the light is received by a phytochrome enzyme or protein inside the cytoplasm, it causes the message to be transducted or transduced in the cell. And usually when there's a molecule that's part of the transduction pathway, we call it a second messenger, because it's like the go-between, it's the in-between. It's going to activate some kinases. And remember, a kinase is an enzyme that phosphorylates, so it usually takes ATP. It takes a phosphate group from ATP, it phosphorylates, okay, and it's activated, and in this case, it's going to activate a transcription factor, and transcription will happen, and then the gene for a specific cell mRNA will be made, and so you guys should remember what transcription is. And if that transcription happens, the mRNA is made, then translation will happen, and then a protein is made, okay? So it doesn't have to be a hormone. It could also be some sort of environmental condition like light. Like in the case of these Arctic animals, we have seasonal coat colors that will change. Like in the winter, coat color or feather color will turn white. And then in the fall, in summer months, they will be brown, okay? And this is an evolutionary adaptation that enables the survival of these species. So how does this happen? Well, first of all, what the animals are doing is they're detecting the amount of light that happens, the amount of light in the daytime. Okay, so if it's short daylight, then the amount of melatonin goes up, and when the amount of melatonin goes up in the brain, then it decreases prolactin. Okay, prolactin is another hormone that influences pigmentation. Whereas if it's a long day, okay, long day, so in the winter time, it's going to be a short day, but in the summer, it's going to be a long day, the melatonin goes down and then it increases prolactin. So in the summer, when prolactin is stimulated, then you're going to see pigmentation in the feathers and fur. They're going to be a darker brown color, okay? And then the lack of pigmentation during a short day, like in the winter. And there's a variety of ways that ligands can be exchanged between cells, okay? The first one is autocrine. You know, the prefix auto means self, and autocrine signaling, this is where a cell targets itself. It sends a message to itself. It's kind of funny how the signal goes out and it comes back in. Okay, so that's autocrine signaling. And then there's also signaling across gap junctions, okay? You know, when you have two cells that are neighbors, you might have these gap junctions, these proteins that are like tunnels that allow for signal molecules to actually travel across the tunnel and go to the targeting neighboring cell. So that's signaling across gap junctions. And then you have paracrine signaling, where a cell targets a nearby cell. And the signaling molecule, or sorry, signaling cell, it sends a signal which will go to a target cell nearby. Okay, and we'll talk about specific examples of all of these in just a few minutes. And then endocrine signaling, this is when a target cell releases a signal to a distant cell, and so the signal has to travel through the bloodstream to get to its target cell. Remember how I was talking about epinephrine is released by the adrenal glands, which are over by your kidneys, and then they go all the way to your head, to your eyes, they go everywhere, okay? And so they travel from the signaling cell to somewhere farther away in your body via the blood. So autocrine, these are just more pictures of what's going on. Autocrine signaling, paracrine signaling, and then endocrine signaling. And notice how the target cell has these receptor proteins, and it looks sort of like a lock and key, doesn't it? Where the signal, the ligand, binds to the receptor protein. Okay, so across gap junctions, an example of this would be cell-to-cell contact with lymphocyte communication, and we'll be talking about that as well today. Lymphocytes are basically the B cells and the T cells that make up your immune system, and I need to move myself out of the way because, let's see here, I don't need that. I need to move myself out of the way. How do I do that? Okay, there we go. So you can see here that this is a macrophage, or a, hold on, let me grab my pen. Okay, so the macrophage here, it's making contact with the helper T cell. So it's like cell to cell, like the cells are touching each other, okay? I don't know what's going on there. Okay. And then across gap, well, you know, this is cell-to-cell contact, lymphocytes, B cells, and T cells. So it's the same thing as the previous slide right here. Across gap junction, cell-cell contact. Across gap junction, cell-cell contact. I'm not sure why I repeated this slide again. And then another example is within plants. Plant cells that are neighboring their cell-to-cell contact, they don't have gap junctions, but we have what we call plasmodesmas or plasmodesmatas, okay? And so the cytoplasm can move across the space, the gap. Okay, and then paracrine signaling, and we'll talk about this when we go over the nervous system, is neurotransmitters. So an action potential or an electrical potential will move down the axon from one neuron, and then it'll get to what we call a synapse. The synapse is like where two neurons are not in contact, but they're almost in contact. They're like in close proximity. And the cell that is sending a signal, it releases neurotransmitters, which are proteins. And those proteins, those ligands, neurotransmitters are ligands, they're going to bind to receptor proteins on the receiving neuron. Okay? Okay, and then long distance signaling, like happens with the endocrine system, production of hormones by glands. So glands are going to send the signal, and they're going to travel through the blood circulatory system to get to the target cell, like insulin and glucagon. All of these examples are important for you guys to know, which we'll go over for sure in this video. Okay, so when we talk about how your body is regulated by communication, it really depends on two systems. There's two systems that go hand in hand. You have the endocrine system, and then the endocrine system and the nervous system. They go hand in hand, okay? The endocrine system, as mentioned before, is a system of glands, a system of ductless glands. So basically what that means is that these hormones are made in the glands, and then they go straight into the bloodstream. They're not like stored in a duct, okay? And so they're secreted directly into the blood, where they will travel to target tissue. And then target cells have the receptor proteins. And the endocrine system is usually a slow but long-lasting response when compared to the nervous system. Nervous system, the way that cells communicate with each other is rapid, quick, fast response, okay? Endocrine response is pretty quick. Like, again, the fight or flight response, I mean, your life's in danger, you got to react quickly, right? But compared to the nervous system, it's slower. And the nervous system is made up of neurons, where the message that goes through a neuron is an electrical signal. It's an electrical signal. And when that electrical signal reaches what we call the axon terminals, a chemical is released, a protein is released, a ligand is released called a neurotransmitter, okay? Throughout this video, I'm going to try to use as much vocabulary as possible. So neurotransmitters are a type of ligand that is released from a neuron. And it's very fast, and it's short-lasting response. So with neurotransmitters, and that's what's going on in this picture here, an electrical impulse is going to move down an axon of a neuron, and when the electrical impulse reaches the axon terminal, like where this axon ends, there are these vesicles that hold these neurotransmitters, and those neurotransmitters will be released by exocytosis. Does that sound familiar to you guys? Okay, and those neurons are going to bind to receptor proteins and cause the target cell to respond. Okay, so that's like the first part of our signal transduction pathway. Remember, the ligand binds to the receptor. And there's our neurotransmitter. And then hormones are released by glands, endocrine glands. So you can see this is the gland tissue. It releases hormones directly into the bloodstream, which go to the target cell, where the receptor, target cell has receptor proteins that bind to the hormones. I'm kind of nervous. I don't know if I'm recording or not. I hope I am. What if I'm not? And I'm wasting time? Let me see. I can't tell. Let me see here. Let me exit this PowerPoint just in case. I just have to check. Does that mean it's recording? I think so. Okay, I think we're all good. Yeah, I think we're fine. Okay, so let's talk about some classes of hormones that are part of the endocrine system, okay? Basically, these hormones belong into two main categories. You have the protein-based hormones. Basically, these are hormones that are made up of protein. They could be, some could be, have a bit of carbohydrates attached to it. We'll talk about that in just a second. Or they could be lipid-based. So it's either protein-based or lipid-based, okay? And you have to think about, what are proteins made up of? What are some characteristics of proteins versus what are lipids made up of? What are some characteristics of lipids? Something to really think about, because this is 100% going to be on the AP exam. You need to know how protein-based hormones act versus lipid-based hormones. And it goes back to the characteristics of proteins versus the characteristics of lipids. Okay, remember proteins are made out of, they're polypeptides, right? Polypeptides because they're amino acid chains. Remember, they're amino acids that are linked together by peptide bonds. And, you know, some examples of small protein hormones is insulin, which many of you have heard about, ADH or antidiuretic hormone. There are also protein-based hormones that are called glycoproteins. These are proteins with attached carbohydrates like FSH, LH. FSH is follicle stimulating hormone, LH is luteinizing hormone, which are both responsible for making eggs in the ovaries and sperm in the testes. You don't need to know what all of these hormones do. But I'm just letting you know that there are like many different kinds of hormones. The last one is the amine hormones, protein-based hormone. Amines is what it sounds like it's amino acid. So these are small proteins because they're just, the basic structure of them are amino acids, like epinephrine. Epinephrine is a small hormone. And then melatonin. Melatonin, I got kind of cut off there. The melatonin is also a hormone that's released by your pineal gland in your brain, and it causes you to become sleepy. And it's the amount of melatonin, there's an increase in release every day when the sun starts to set. So once the sun sets and it gets darker outside, you start to release melatonin, which makes you sleepy. Hopefully you're not sleepy right now. Wake up. Okay, lipid-based hormones. We have our steroids. Okay, remember steroids are non-polar, right? They're non-polar. And remember, they are not going to interact with water. And this is going to be really important when we go over how cells, how these hormones get to the cell. Okay, and some examples of steroid hormones are the sex hormones like testosterone, estrogen, progesterone, aldosterone. Okay, so how do hormones act on target cells? And like I said, it has everything to do with the type of hormone we're talking about. Protein-based hormones versus lipid-based hormones. Lipid-based hormones, remember, they're hydrophobic, non-polar, they're lipid-soluble, meaning they mix with lipids, not with water. So remember the cell membrane. Okay, the cell membrane is made up of phospholipids. So let me just try my best to quickly draw a phospholipid, remember the phospholipid bilayer? Okay, so the phospholipid bilayer is primarily non-polar because of the tails, So this section of the phospholipid bilayer is hydrophobic or non-polar. It's only the heads that are facing outside the cell. So let's say this is outside, this is inside. That's polar. And so lipid-based hormones, because they're hydrophobic or non-polar, they can simply squeeze through the phospholipid bilayer and come in. Okay, they can diffuse across the cell membrane and enter cells. They don't need to bind to receptor protein that's embedded in the phospholipid bilayer. All lipid-based hormones, like those sex hormones that we talked about, testosterone, estrogen, progesterone, they can all come in through the phospholipid bilayer because they are hydrophobic or nonpolar. And so what they do, though, is they do bind to receptor proteins that are in the cytoplasm, inside the cell, or they can bind to receptor proteins that are in the nucleus. And when they bind to the receptor protein and the hormone together, they act as a transcription factor, okay? And remember what transcription factors do? They cause transcription to happen, right? And so when transcription happens, mRNA is made, which is going to be translated later on by the ribosome to make the protein. So you guys, basically these lipid-based hormones, these steroids, they're going to act ultimately as transcription factors to turn on genes, And we'll talk about an example of that later. So we turn on genes. protein-based hormones, remember proteins are amino acids. You have like the amine group, you have the carboxyl group, you have R groups that could be polar, making the protein hydrophilic. And so because they're hydrophilic, they cannot simply bypass this nonpolar region of the phospholipid bilayer. They cannot, okay? So they can't diffuse across the cell membrane. So what they have to do is they have to bind to a protein that's embedded in the phospholipid bilayer. And so once, and I did talk about it earlier, but once let me use a different color. Once the ligand binds to a protein, let's say this is my protein, receptor protein, then the receptor protein is going to change shape. It changes conformational shape, and then a message is brought inside. Because it changes shape, it triggers cytoplasmic molecules to be activated, okay? And we're going to be talking about what examples of some of them. So it triggers a secondary messenger pathway because the messenger, the ligand, doesn't get inside, but it's a messenger inside that transmits that information. I hope that makes sense. That's why it's called a secondary messenger system, because it's not the first messenger, it's not the ligand, it's the second one, okay? And it's going to activate internal cellular response. The cell is going to do something based on the signal coming from the outside, okay? So action of lipid steroid hormones. So let's say that, you know, the endocrine gland, it releases a steroid hormone. The steroid hormone, and I apologize about all this noise. Is there a way to like, okay, well anyways, a steroid hormone is going to simply bypass the plasma membrane because it's able to, right? It's nonpolar, it can cross the cell membrane. It's going to bind to a receptor protein. This is our receptor protein that's in the cytoplasm. And then together, this steroid receptor protein complex is going to enter the nucleus and act as a transcription factor. And it becomes a transcription factor making mRNA. The mRNA will exit the nucleus, go to the ribosome, where the ribosome is going to make the protein, and then the protein will be secreted outside the cell. Okay? So the action of this steroid hormone is for the gene to be turned on, and it's going to make this protein. So, I mean, think about like testosterone, for example. Testosterone is like the masculine hormone, and it causes like facial hair growth, right? So it can target the follicle cells on your face, and it causes the hair follicles to produce hair. You know, bone mass is, you know, it can increase bone mass, it can increase muscle mass, right? Testosterone, some people take synthetic testosterone to, you know, bulk up, build more muscle mass. And this is the way that it occurs. I mean, the hormone acts as a transcription factor and it turns on genes, okay? So yeah, this is another picture showing that a steroid hormone, it crosses the cell membrane or plasma membrane. It binds to receptor protein. They enter the nucleus where they bind to the DNA gene. Transcription takes place. mRNA is read by the ribosome, and then you get a new protein. So testosterone causes facial hair growth, like this picture of Paul Rudd. Okay, whereas protein hormones, because proteins are hydrophilic, okay, they cannot simply bypass the cell membrane. They have to bind to a receptor protein. The receptor protein will change shape, okay? And as the receptor protein changes shape, it's going to activate cytoplasmic molecules. Okay, inside the cytoplasm, a molecule, like in this case, this is called a G protein. And the reason why this is called a G protein is because it's energized by GTP. Remember, GTP is guanine triphosphate. It's a type of nucleotide, just like ATP, adenosine triphosphate. It's a type of nucleotide that provides energy because they have 3 phosphate groups, okay? There's GTP, ATP, CTP, TTP. I mean, technically, they can all provide energy inside of a cell, but evolutionarily speaking, ATP is the main one. And in this case, for the G protein, it's activated or energized by GTP. And so that G protein, it actually will come off of this receptor protein. And I almost forgot to mention this, but this receptor protein has a specific name. It's called a G protein linked receptor, GPLR. I forget if I think that might be I'll have to double check, but I think that is the name of this protein. It's a G protein linked receptor because it has a G protein attached to it. But once the receptor protein changes shape, the G protein comes off of it, it disassociates from the receptor protein, and it actually moves laterally, and it activates this enzyme. This enzyme is called adenyl cyclase, okay? And once this enzyme is activated, this enzyme is going to break down ATP. So it's going to take ATP and it's going to cut it, okay? It's going to break it. And it's going to remove two inorganic phosphates, okay? So two inorganic phosphates are going to come off, and you're going to end up with not ATP, but what? AMP, okay? And specifically CAMP is going to be made, okay? And CAMP is going to be used to activate other molecules like kinases, okay? And this is what we call the transduction portion right here, because it's inside the cytoplasm where all of these things are being activated, which eventually is going to lead to the cell responding in a specific way. And we'll go over an example right now. Oh, I almost forgot. CAMP, it acts as a second messenger because, you know, it's transmitting that information, the action into the cell. It's going to cause a cell to respond. The signal protein is not coming in, but the CAMP is like the second molecule that's causing the cell to respond, okay? So epinephrine. Adrenaline, remember it comes from the adrenal glands that sit above your kidneys. So that's the adrenal gland. It releases epinephrine or adrenaline, and you can see these blood vessels attached to it. So they're going to travel through blood vessels all through your body, okay? Epinephrine causes conformational change of the receptor protein. The G protein is going to disassociate and come off of the receptor protein, and it's going to activate the adenyl cyclase. I hope that noise isn't too loud. Adenyl cyclase is an enzyme that breaks ATP, makes CAMP. CAMP is a second messenger, which is going to activate protein kinase A, which protein kinase A is going to activate phosphorylase kinase. And then it's going to activate glycogen phosphorylase. And that is all of that transduction portion. And what glycogen phosphorylase does is that it takes glycogen. And right now we're talking about the liver cell, the target. The target that came from, the target of this is the liver cell. And the liver cell, remember, it stores glycogen. And it's going to cause the glycogen to break down and release glucose into the bloodstream. Okay? What's going on there? Let me just move my face. Yeah. It releases the glucose to the blood. Okay? And if you think about it, right, if this is all part of the fight or flight response, your life's in danger, you need to either fight back or you need to take flight, which is to run away. You're going to need that glucose. Aren't you? You're going to need that glucose, that energy that's going to, remember, cellular respiration breaks down glucose to make ATP. You need that glucose to make ATP energy so you can either run away or you can fight back when your life is in danger. It all makes sense. Okay, and that epinephrine, it's going through the bloodstream, that adrenaline is going through the bloodstream, and it targets several cells, right? Like it could target the liver, and it causes the liver to eventually take that glycogen, breaks it down, and glucose is released from the cell. Your blood glucose levels are going to go up because that is going to be an energy molecule, energy source to make ATP. Whereas if the target cell is a smooth muscle cell, okay, smooth muscle cell in the wall of blood vessels that supply skeletal muscles, that epinephrine is going to bind to the receptor protein, and it's going to cause the cell to relax. And when the cell relaxes, the blood vessels, they don't constrict, they actually relax and open up. The blood vessels open up and increases blood flow to the skeletal muscle. And you want that to happen because, again, if your life is in danger, you need blood flow to your skeletal muscles. Those are the muscles that are attached to your bone to move, to run, to fight, okay? And then the target cell, epinephrine, can also target smooth muscle cells in the wall of blood vessels that supply your intestines. Okay, remember we talked about at the very beginning how in the fight or flight response, it slows down your digestion. And how does it happen? Epinephrine binds to the receptor protein on the blood vessels that supply intestines, and it causes the cell to contract, and the blood flow will, the blood vessels contract, and it decreases blood flow to your intestines. And so as a result, when blood vessel constricts, it decreases blood flow to the intestines, and your intestines are not going to be energized to do what they need to do, which is digest your food. And it kind of makes sense, right? Because don't you want blood to flow to your skeletal muscles rather than your intestines when your life's in danger? You'd rather have blood flowing to your skeletal muscles than your intestines, because when your life's in danger, your body doesn't want to be focusing on digesting food. It wants to be focused on fighting or taking flight. Okay. So part of the whole second messenger system is what we call a cascade. Okay. So we have one signal molecule or one ligand. One ligand can cause a very fast response within the cell because that one ligand can actually activate multiple G proteins. Okay, like the response is going to be amplified. It's called amplification. Okay, amplification, because that one signal is going to activate, like in this picture, 3 G proteins, 1 to 3 G proteins, and each G protein is going to activate 1 adenyl cyclase, and then each adenyl cyclase is going to be able to activate like 8 CAMP, and then each CAMP activates a protein kinase, and then each protein kinase activates several enzymes, which is going to produce many products. So, and it all happened from that one signal. So amplification, going to cause a huge, fast response, okay? Cascade multiplier, because starting from 1 and you multiply by 3 and then you multiply by 8, and it's just a very fast response. Okay, kind of moving gears here, because we're going to start talking about specific examples in the body system. Maintaining homeostasis, why is it so important? You remember, homeostasis is being able to maintain stable internal condition, so a specific body condition. For example, you know, your body temperature should be around 98.6. If it gets too hot, or if it gets too low, below that, then your body is going to react in such a way that it wants to bring it back to its normal homeostatic level, okay? So if it gets too high, a gland, like we're talking about the endocrine system, is going to release a hormone. That hormone is going to go to a target cell, and it's going to lower the body condition to being a normal homeostatic level. If it gets too low, then a gland is going to release a different hormone, and it's going to bring it up. It raises the body condition, so it's back to a specific homeostatic body condition. Okay, your body is like, it's fighting to keep it at that same level. Okay, so let's talk about some examples of homeostasis. Why is it so important? Well, I mean, our cells are sensitive to changes. Like, remember your enzymes that control chemical reactions? Aren't they sensitive to changes in pH, in changes in temperature? Right? We need to keep everything at a specific condition, or else those enzymes are going to I hope you guys said denature, right? So we need to keep our body at a specific temperature, specific pH, or else those proteins and enzymes are going to denature, okay? And this is what we call the negative feedback model, or the number 8 feedback, okay, loop, all right? If it gets too high, it brings it back down. If it gets too low, it brings it back up. Okay, a good example of this is controlling body temperature. Like I said, your body wants to keep the body temperature about 98.6 degrees Fahrenheit or 37 degrees Celsius. If it gets too hot, like your PE teacher makes you run 2 miles in like 100-degree weather. Hopefully they never do that. Your hypothalamus, there's blood flowing through the hypothalamus. There are sensory neurons that will detect how hot your blood is, okay? And if it's too hot, like it's above 37 degrees Celsius, then it's going to send a message. It's going to send a nerve signal message to your sweat glands to start sweating, okay? Do you guys like that picture? And it's going to cause the blood vessels just underneath your skin to dilate, because when the blood vessels dilate, they get wider, they get bigger, and it releases body heat, that blood heat to the surface, and all that body heat gets lost at the surface of your blood. So, I mean, you guys see this, like when you guys, oh my goodness, like after PE, right? I have students coming into the classroom and they're sweating and you could just feel the body heat coming from their body, okay? And then if your body temperature gets too low, the hypothalamus, again, can detect the change in temperature, sending nerve signals to your muscles to start moving. Because when your muscles start moving, right, you start shivering, then you warm up. And then it also causes your blood vessels to constrict. So it gets super skinny. The blood vessels get really skinny. It decreases blood flow. Why does this happen? Well, because blood carries body temperature, right? And you don't want to lose that body temperature to the outside, to the environment. You want to keep your body core, that holds all of the important organs. You want to keep your body core as warm as possible, but your skin, I mean, it gets cold, but at least your organs are safe, okay? And so it's going to constrict surface blood vessels. And so, you know, some people that like hike all the way up like Mount Everest, it's so cold that the blood constricts so much for so much of a prolonged time. You know, they get frostbitten, right? Their nose, the tip of their nose, the tip of their ears, fingertips, toes, frostbitten, and they need to get amputations, right? And so that's what happens when there's constricted blood flow to those, you know, regions for a long period of time. Then those cells, the tissue there is not going to get the oxygen needed to stay alive. The tissue is not going to get food, glucose needed to stay alive. And so that's what happens with getting frostbitten, or when you get frostbitten, you might have to get an amputation. Okay, this one you 100% need to remember. I see this example all the time, even in some of the practice exams. You guys will definitely address blood sugar regulation and the 2 hormones that are responsible for blood sugar regulation, okay? You know, our blood sugar level, the homeostatic level, should be about 90 milligrams per 100 mils at a fasting state. So when you go to the doctors, they might say, oh, we need to do a blood draw. Don't eat anything for 12 hours. They want to take your they want to measure your blood sugar levels at a fasting state. And it should be around 90 milligrams over 100. It shouldn't be over 100 milligrams per 100 mils, okay? However, let's say that, I mean, your blood sugar levels are going to go up when you do eat something, okay? So when you eat food, let's say you had your Lucky Charms cereal for breakfast or Cinnamon Toast Crunch cereal for breakfast, your blood sugar is going to go sky high, right? It's going to go sky high. Well, the pancreas is a gland in your body that detects this increase in blood sugar levels, and it's going to release a hormone called insulin. We've all heard of this hormone. And the insulin in your blood is going to target specific cells in your body. It's going to target your body cells to take up the blood, take up the sugar that's in your blood. And the sugar is going to go into your body cells, being removed from the blood, and that's how your blood sugar levels go back down, because the sugar is being taken in by cells. It's no longer in the blood. And that sugar is useful, right? Because it's going to be used for cellular respiration. It's going to target body cells to take up sugar from blood. It's also, like, if you have excess sugar in your blood, it's going to target your liver cells to store the glucose away as glycogen. And then also reduces your appetite. Okay? Now, let's say that your blood sugar levels go too low. Like, let's say you're fasting. Okay? Then the pancreas, same gland, it's going to detect this, and it's going to release a different hormone. And that hormone acts antagonistically or oppositely to insulin. It's called glucagon. Okay? Glucagon targets the liver to release glucose. Remember how we store glycogen in our liver? Well, now the liver is going to break it down, the glycogen, and release the glucose into the blood, raising blood sugar levels back to homeostatic level of 90 over 100 mils. And then you might get hungry. Glucagon is going to trigger hunger. Okay? Everybody has heard of insulin, right? Very few people have heard of glucagon. But one way I remember this is like, we're talking about glucose, right? Sugar. So glucagon is like glucose gone. You like that? Okay? It's like glucose gone. When glucose is gone, the levels are low, then glucagon is released so it can bring back the glucose levels. Okay? I'm so nervous that I'm not recording. Let me escape out of this. Discard. Hold on. Yeah, I think I am recording. Okay. Let's skip the calcium stuff because it's not that important, but this is just another example of two different hormones, actually more than two hormones, that regulate blood calcium levels, regulates homeostasis within your body, okay? Yeah, there's actually several hormones involved with this one, like calcitonin, parathyroid hormone. Okay? Okay, we'll go over this diagram, but this diagram has to do with what we call the thyroid hormones or the thyroxins, okay? You know, thyroxins and thyroid hormones are really important because they are responsible for so much. I mean, if you take a look here, on this list, okay, the thyroid, it produces thyroxine hormones, and it's responsible for metabolism and development, for bone growth, mental development, metabolic use of energy, blood pressure, heart rate, muscle tone, digestion, reproduction. It's responsible for so much. Because your thyroid produces two kinds of hormones. The thyroxine hormones are the T3 and the T4. Those are the thyroxine hormones. If you make too much, it's called hyperthyroidism. You may have heard of hyperthyroidism. Like hyper is like above, right? Like you're making too much of the homeostatic level. Or you may have heard of hypothyroidism, and that's when it's like underperforming. It's under. Hypo is under. You're not making enough, okay? Well, these thyroxins, if you make enough of them, okay, so I should go back to this, right? This is we call the pathway to making the thyroxins, to making the T3, T4. It starts here in the hypothalamus. The hypothalamus is a gland in your brain that makes several hormones, like TRH hormone. It makes TRH and it releases TRH. TRH is TSH releasing hormone. TRH, it targets, remember we're talking about like a gland releasing to a target cell. It targets the anterior pituitary. And the anterior pituitary is also in your brain. And the anterior pituitary, it stores TSH. TRH is going to cause the anterior pituitary to release TSH. TSH released from your brain goes all the way down to your thyroid, which is over here, causing the thyroids to release the T3 and the T4, okay? But if you are already producing and releasing enough T3 and T4, then it's going to, by negative feedback, and my head is kind of, you know, blocking it, but there's like a negative sign here. It's negative, by negative feedback, it's going to cause the hypothalamus to stop releasing TRH. That's negative feedback, right? Negative feedback stops the process from happening. Or if TSH, if there's a lot of TSH in your bloodstream, then it's going to act by negative feedback on the hypothalamus, stop the hypothalamus from producing TRH, okay? So again, these are parts of your body that is sending messages that are going to help maintain homeostasis so that you can develop properly, everything is happening normally, okay? So this is a pathway that you need to understand, like, you're going to see on the AP exam, like, all these arrows and, like, we'll talk about, like, the T-shaped line. So see how there's, like, a negative sign here? Sometimes they don't draw it this way. Sometimes they'll, like, draw it, and let me use a different color so it kind of stands out. Sometimes they'll draw a line that goes like this, and instead of an arrowhead, it'll be like a straight line. And that straight line, it's like a T-shaped line, means that it inhibits the hypothalamus, okay? But you need to be able to interpret diagrams like this. And the hypothalamus, as I mentioned, is part of your brain. It's the master nerve control center. Like, it detects your body temperature. It detects the amount of, like, hormones or solutes in your blood. It does so many things, okay? And then the pituitary gland is a master gland because it holds so many hormones, and it secretes a broad range of tropic hormones or tropic hormones that causes other glands in your body to release other hormones, okay? And I feel like we don't have time to go over this, and I don't think it's that necessary, okay? But like in your brain, and this is supposed to be kind of like an excerpt from your brain, you have the hypothalamus, that's like the master control center, right, in your brain. And it's connected to the anterior and posterior pituitary gland. Remember how we said that the pituitary gland is a master gland? Well, it's called the master gland because it stores so many different hormones. It stores TSH, stores and releases TSH, ACTH, growth hormone. I know I've talked about growth hormone with my students about how growth hormone causes somebody to grow taller. And so you can actually take growth hormone like Lionel Messi did, right? They predicted that he would be super small, but then he took growth hormone, which increased his height to several inches. You have FSH, LH, prolactin, MSH, and then you have oxytocin, my favorite. If there ever was a favorite hormone, it would be oxytocin because it's called the love hormone, okay? I mean, it does a lot of other things. Like, for example, when a woman goes into labor, oxytocin, it targets the uterus to start contracting and pushing the baby out. That's not why it's my favorite, obviously not why it's my favorite, because it causes a lot of pain. But it's also the love hormone because when you fall in love with someone, the amount of oxytocin in your blood is very high, and it gives you the feeling of love and trust. Like, when people get into a relationship, you can actually measure the amount of oxytocin in your blood abnormally high, higher than usual. And it is responsible for the feeling of trust and love. Like, you feel all the warm fuzzies. That's due to oxytocin. And couples that have been together for more than 2 years, the amount of oxytocin goes back down. So you might not feel the love and the fuzzy feelings after a couple of years because you can blame it on the lack of oxytocin. Okay, but again, you know, we're humans, and we there's, you know, it's not just about feelings, right? We have also trust, respect, and there's a lot more involved in relationships than the warm fuzzy feelings, right? And then you have ADH, antidiuretic hormone, which targets the kidneys to conserve water. Okay, let's move on. I feel like we've already gone over this, so let's skip the next few slides. I'm just going to escape out of this PowerPoint so you guys don't need to hear all of that. Okay, let's talk a little bit about protein hormones now, or receptor proteins. Just want to make sure that I'm recording. I'm so paranoid that I'm not recording, but I think I am. Receptor proteins. There are different kinds of receptor proteins. There's a huge diversity of structures, but here are some general features about these receptor proteins that bind to ligands. There's going to be one area of the protein that is where it interacts with the ligand. So adrenaline binds here to the receptor protein. So there's an area that interacts with the ligand. And then there's they're inside the receptor protein that transmits the signal to another protein, because this part right here, it changes shape. It changes conformational shape, okay? So an area of the protein that transmits the signal to another protein, the signal transmission is accomplished through conformational change. So you guys, this is actually what a G protein looks like. And it's attached to the receptor protein, and when the receptor protein changes shape, it causes the G protein to change shape, be activated, and then it's going to activate adenyl cyclase. And remember what adenyl cyclase does? It breaks ATP. It breaks off two inorganic phosphate groups from the ATP and makes CAMP, okay? So there are 3 types of signal receptors that we're going to go over, but there's way more in actuality, okay? There's the G protein linked receptors, the GPLRs, there's the tyrosine kinase receptors, and then there's the ligand gated ion channels. Again, this is the G protein linked receptor, sorry, what did I say? GPLR, okay? Because it's linked to a G protein, and that G protein is going to activate adenyl cyclase, which is the enzyme that's going to break apart ATP into the second messenger called CAMP. I'm talking super fast. Okay, and there's a tyrosine kinase receptor, okay? I'm seeing more and more of these pictures of tyrosine kinase receptors, but it's actually a dimer, okay? A tyrosine kinase receptor is actually made up of two proteins, okay, that come together once there's 2 signal molecules. The signal molecules, when they bind to each half, then the 2 halves come together, and the reason why they're called tyrosine is because, again, proteins are made up of amino acids, and tyrosine is a type of amino acid, and those amino acid tyrosines are going to be phosphorylated. 6 ATP is actually going to be used here because there's 6 tyrosines. They're going to be phosphorylated. Once they become phosphorylated, they activate another enzyme called phospholipase C. And this enzyme is going to take a special phospholipid. This is a phospholipid embedded in the phospholipid bilayer called PIP2. PIP2 is going to split into 2 pieces. One is DAG. The other is IP3, and in this case, you guys, IP3 is a second messenger because it's in the cytoplasm, and it's going to act as a messenger in the cytoplasm, and it's going to cause the cell to respond. So here's IP3. IP3 binds to the smooth ER. Is it the smooth ER? Yeah, it's a smooth ER. It binds to an ion channel in the smooth ER. And do you guys remember the smooth ER not only makes lipids, but the smooth ER also stores calcium ions, okay? And that's where calcium ions are stored. And once IP3 binds to the ion channel of the calcium ion channels, it opens up, calcium ions rush out, they bind to another protein called calmodulin. And calmodulin, depending on the type of cell, has various actions. It does a lot of different things. It has a diverse list of things that it does. So that's a tyrosine kinase receptor protein, okay? Whereas the ligand-gated ion channels, the ligand binds to the ion channel, causing it to open up, and then the ions either come in or leave, depending on the action, okay? And it triggers a cell response. Okay, we're going to skip that. And here's a really interesting diagram showing you, the pictures I've showed you so far of signal transduction pathways where a ligand binds to receptor protein, they were very simple, but in actuality, you guys, signal transduction pathways are very, very complicated. I mean, look at this. There's many different kinds of receptor proteins, first of all, And depending on the signal molecule, there's going to be a specific action that's happening within the cell, right? We're going to be talking about hedgehog later on in this lesson, but there's a lot of signals that's going to cause the cell to commit cell suicide or apoptosis, okay? All right. I'm going to skip this one because we already went over this, but basically the benefits of a second messenger system is, you know, you have this multiplier amplification system that's happening, okay? Here's a practice question that was released by the College Board, okay? And believe it or not, this is a G protein linked receptor pathway, okay? So epinephrine, which we talked about too, is a protein hormone, right? Because it's a protein hormone, it cannot simply bypass the cell membrane. It needs to bind to this G protein linked receptor. This is the G protein. It has several subunits. It has the alpha, beta, gamma. It has 3 subunits. So thinking back to protein structure, do you guys remember? You have the primary structure of protein, the secondary, tertiary, quaternary. If you have 3 subunits, alpha, beta, gamma, what level of protein structure is a G protein in? Hopefully you said quaternary because it has, you know, quaternary means 2 or more tertiary structures coming together, okay? So what happens here is the alpha and the beta, they come off, and it's the, the gamma and the beta, it disassociates from the alpha. It's the alpha subunit that activates the adenyl cyclase, and there's this, all these arrows mean that it, you know, it's activating, so forth. Okay. Which of the following outcome will most likely result from the irreversible binding of GDP? I need to move my head to the G protein. So what's happening is GDP, it's saying that it's stuck to the G protein like this. And when it's stuck to the G protein, GTP cannot bind to the alpha unit. And if that happens, what most likely is going to be the result of this? Hopefully you're thinking, well, if the G protein is stuck and the alpha subunit can't come off, then none of this is going to happen, right? None of that's going to happen. And so the answer is going to be A, the intracellular concentration of glycogen will increase. Because remember, with this whole pathway, what happens is the end result is that glycogen is broken down into glucose, and then it can be used for cellular respiration or glycolysis, which is the first step of cellular respiration. And so instead of all of this glycogen being broken down, it's just glycogen is going to remain, or the concentration is going to increase as glycogen is continuously stored, okay? And then you might see on the AP exam a diagram like this. Remember I was saying how you have the arrowhead. Arrowhead means activation, activates, but then the T-shaped line means it inhibits. So, you know, which pathway? First pathway versus second pathway, you know, which pathway activates or stimulates cell division? And hopefully you're thinking of the first pathway. Second pathway, it doesn't stimulate cell division because GPIP, it inhibits the G protein. It inhibits the G protein, okay? All right. So again, what do the arrowhead lines and the T lines represent? Arrowhead means activates, whereas the T lines represents inhibition. Okay, it stops. Okay, so now we're going to talk about animal form and function. Whew, I need a breather. So when we look at the form, like the physical body form of animals, they're over evolutionary time, their body shape is constrained by physical forces, okay? Like for example, in this picture, you have 3 very, very different animals. You have a mammal in the form of a seal, and then you have a bird in the form of a penguin, and then you have a fish, that's a tuna. Tuna in the form of a fish, but their body shapes are very similar, and that body shape is actually called fusiform. It's like hydrodynamically an advantage to have that type of body form so they can glide and swim through the water. This is an example of convergent evolution. So they've independently, their ancestors independently went through mutations that gave them this body type, this body shape, and it's an advantage, right? It's a selective evolutionary advantage to have that body shape, so it enabled them to survive, and it enabled them to survive and reproduce and pass along this body shape to their offspring, okay? And when we look at larger organisms, we see this evolutionary trend where if we increase body size, we have to increase surface area for nutrient absorption, okay? Versus, I'm going to go back to this picture in just a minute, versus a single cell organism like this amoeba. An amoeba can easily get nutrients and oxygen from its surroundings by diffusion. Simple diffusion, oxygen going in, carbon dioxide waste going out, nutrients coming in. Whereas if you're a bigger animal, like let's say, you have your heart tissue in the middle of your body, how is oxygen going to get there? How is food going to get there? It's got to go through the circulatory system, okay? Oxygen goes in, you breathe it in through the respiratory system, and your respiratory system has it's a branched organ with lots of branches called alveoli to increase surface area where you can do very efficient gas exchange. And then also your digestive system is made up of a very long tube. The surface area is increased by having even the internal surface of the intestines be very bumpy. And if you have more bumps and lumps and more, folds, highly folded internal surface, then you're going to increase surface area so you can actually absorb more food. Like I think our textbook mentions how our digestive tract, the intestines alone, has a surface area equal to a tennis court, right? Because we're trying to absorb as much nutrients as possible. But if you're a single cell organism or you have 2 layers of tissue, simple diffusion is how you would get oxygen and nutrients. Okay, and then animals, we have to maintain homeostasis. We do that by feedback regulation. We already talked about that in terms of like body temperature. So we're going to skip this. What are some ways that animals regulate body temperature? Okay, well, first of all, some animals have insulation like fat, feathers. When I say fat, I mean like blubber, like in whales, seals, insulation, in the form of blubber, and then fur, feathers, okay? Vasoconstriction and vasodilation. So I'll show you a picture in just a minute. Vasoconstriction is the blood vessels constricting, getting more narrow. Vasodilation, blood vessels are getting wider and dilating. And then you have countercurrent heat exchangers, which we'll go over. You have evaporative heat loss, which is going to cool you down, right? If you sweat, and the liquid water evaporates off of your skin, you're losing, the water vapor as it turns into, as the liquid water on your skin and your sweat, it turns into gas. It's absorbing that heat energy and turning into a gas, and it's leaving your body, and that's how you cool down. And then behavioral responses. Animals can go in the shade when it's too hot. Some animals will lick their skin, so because the saliva has water and they can cool down by evaporative heat loss. Elephants will splash water on them, or pigs will roll around in the mud to cool down. So those are some behavioral responses that are going to help maintain body temperature. And then there's also change in metabolic heat production. We'll talk about that too. Like an example of that is hibernation is an example. So this is what vasodilation is when you get too hot. The blood vessel just under your skin, they dilate, they get larger, and so the blood, as it flows to your skin, a lot of that heat is lost at the surface. Whereas vasoconstriction, it reduces blood flow and heat transfer by decreasing the diameter of the blood vessels just under your skin. Okay, and then the counter-current heat exchanger, you can see this in animals that are warm-blooded, but they live in cold environments and they don't want to lose the body heat that's inside of them to the outside. So what happens is that the blood vessels so I should say veins, right? Veins carry blood back to your body, like to your core, like your heart, whereas arteries deliver blood to the extremities, okay? So blood is going to leave the heart through the artery, like in this direction, and then blood is going to flow back to the heart through the vein. And so, you know, when the blood is being carried away from the heart, it's warm blood, right? And if that warm blood reaches the surface, near the surface where it's very cold outside, then heat's going to be lost to the outside. So what happens instead in countercurrent heat exchange is that the artery and veins are almost touching each other. They're side by side, and the heat is going to transfer over to the vein. So as the blood is flowing out, heat transfers to the vein, because as the blood is coming back into the body through the vein captures that heat energy, and so all that heat energy never reaches the outside. The heat energy is maintained inside the body, close to the core of the body. I hope that makes sense. And same with the dolphin. Dolphins are mammals like us, warm-blooded. So as the blood flows to the tip of the fin, heat moves over. It moves over to the vein so that heat is not lost at the tip of their fin. Okay. Okay, and then yeah, so we can skip this one. And I was talking about how like hibernation is a way to maintain body temperature, because if you think about animals that hibernate, animals that hibernate are usually found in very, very cold ecosystems. Like this is a Belding squirrel. Okay, this is a picture of a Belding squirrel, and they live in the Sierra Nevada mountains, like Yosemite area, where it's like it gets super cold in the winter. And so in the wintertime, food sources are very scarce. There's not a whole lot of food around. So if they were awake during the winter time, they could possibly die because they're out in the cold, there's no food, they're trying to stay warm because they are warm-blooded. They're going to expend a lot of energy, starve to death probably. Okay, so instead of being awake in the wintertime, they'll actually be in their burrow underground where it's warmer, and they'll be asleep. They'll be hibernating. Okay, and then it's kind of funny because they hibernate for such a long time. If you look at this diagram, their body temperature goes down. Okay, starting in early October. Okay, oh no, is that September? Yeah, this is September. So in September is when their body temperature goes down, because that's when they are hibernating. So they start hibernation in September, and then they wake up in May. Okay, in May. So that's a long time. Okay, they're like sleeping for half the year. Would you guys like that? Would you guys like to sleep for half a year? They do have moments of arousal. Arousal is where they wake up for a minute and then they go back to sleep. And they do this like on a weekly basis. I think it's, yeah, weekly basis. It's sort of like a car where you got to start it up. If it's just sitting in your driveway and parked, it's going to die. The batteries are going to run down. But if you like start the car once a week, then the batteries will stay alive. Okay, so it's sort of like that, where the body temperature goes down and then it'll go up for just a moment and then it'll go back down. So their body temperatures can get really, really, really low. Like in this diagram, it's about, I would say, so the lowest point is right here. What temperature is that? Like 2.5 degrees Celsius. That's almost freezing, right? Because 0 degrees Celsius is freezing. So if they don't have to spend a lot of energy staying warm, they conserve energy. They can stay alive during those difficult months of winter, and then they'll wake up when it's nice and toasty and warm outside and there's plenty of food. Okay? So sources of body heat, there's two kinds of animals, right? There's the endotherms. Endo means internal, therm meaning heat, internal heat, like this bobcat. Us, we are endotherms. We're like warm-blooded. We don't really use that term warm-blooded that much. We want to use the word endotherms, okay? Or ecto means outside, outside warmth, okay? Like a snake is an ectotherm. Their body temperature relies on their environment. They do not maintain internally, they don't maintain the same body temperature. They need to like bathe in the sun to get warm, okay? Or if it gets too hot, they need to burrow under a rock or something. They need to go somewhere in the shade to stay cool, okay? A bobcat or us, an endotherm, our body temperature is pretty much the same. It's very stable, homeostasis, it's stable. No matter what temperature it is outside. It could be like 0 degrees outside. It could be 30 degrees outside Celsius. But our body temperatures will stay pretty much the same because we maintain we're endotherms, okay? Whereas a snake, their body temperature changes with whatever it is outside. If it's like, you know, almost 30, if it's 30 degrees outside, its body temperature is 30 degrees. If it's 35 degrees outside, its body temperature is 35, okay? So, you know, there's 4 processes that account for heat loss or gain. There's conduction, convection, radiation, and evaporation. This stuff I think we can skip. But it is, I think what's important about this diagram, it shows you that, you know, ectotherms, they don't spend a lot of energy, you know, they don't spend a whole lot of energy, ATP, when they're at rest. Okay, so this is the alligator. If you look here, their SMR is the standard metabolic rate. This is the metabolic rate when they're doing nothing. They're just resting. Okay, for a human, our resting metabolic rate is a lot higher. I mean, compared to an alligator. Because we're endotherms. We have to spend energy staying warm. Whereas alligators or ectotherms, they don't have to spend energy staying warm. Okay, and I think this diagram is a good one. I just gotta move my head. So notice how, like with this python, if you look at this pie chart, the orange piece of the pie is missing from the python because they're reptiles, they're ectotherms. They don't need to spend energy maintaining body temperature, temperature regulation costs. Whereas we do, so do birds and so do other mammals like this mouse, okay? Okay, I'm just going to press pause here and just take a little water break. Turns out that I've been recording for like an hour. Yeah, not too bad. Okay, so now we're going to move on to the immune system because, I mean, I feel like you don't need to know like every single detail, but I feel like it's better to be over-prepared than under-prepared, right? So that's why this video is going to be long, and that's why my PowerPoint is so long, is because I don't want to miss anything. I want you guys to have all the information so that at least you're over-prepared. But again, the whole like big picture of what we're talking about is how cells communicate with each other, okay? How cells communicate. And your immune system is completely dependent on communication. You know, your immune system defends your body against pathogens and foreign invaders, and cell communication is vital for the immune response, all right? You have several lines of defense. Okay, several lines of defense against invaders. There's something called innate immunity. Let me grab my pen. There's something called innate immunity. And innate means what you're born with. Okay, it's what you're born with. It's a rapid response to a broad range of microbes. And there's 2 ways in which you have innate immunity. There's the external defenses, like keeping viruses or bacteria, foreign invaders outside, keeping them outside, okay? Your skin is such an important barrier, right? It prevents infection. Mucous membranes, you know, you produce mucus in your mouth, in your nose, Your respiratory tract produces mucus because your mucus is sticky, and when you breathe in microbes, they get stuck on the mucus, and then you like blow your nose or you cough it out, and then they're out of your body, right? Secretions like sweat, acids from your skin, okay, they protect you from viruses, okay? And then let's say that a virus does get into your body, okay? Then there's internal defenses like phagocytic cells. There's also antimicrobial proteins. There's the inflammatory response. And then you have something that sounds really like cool, or the natural killer cells, okay? And these natural killer cells are a type of macrophages or phagocytic cells that go around and then they like kill the microbe, or sometimes they can kill your cells that are infected, okay? And then so that's innate immunity, like what you're born with. And then there's acquired immunity. That's what you gain in your lifetime. This is what you gain in your lifetime when you are exposed to microbes, pathogen, when you are exposed to a vaccine that mimics the invader, okay? And the acquired immunity, there's actually two parts to it. There's the humoral response, which is primarily antibodies that you create. These are proteins. And then you have the cell-mediated response that has to do with the T cells, okay? Like the cytotoxic T cells or the helper T cells. So we're going to talk about the immune system now. Like I said, first line of defense is innate. The innate is something you're born with, okay? Like the physical barrier, like skin, hair, mucous membranes, and urination. Like when you go, when you urinate, you're also urinating out any bacteria that are near that part of your body, okay? There's the chemical line of defense. You know, your skin is pretty acidic, okay? It's 5.5 pH. 5.5 pH is acidic and it can kill bacteria. Your gastric juices, like your stomach has fluid or juices, stomach fluid, that's very, very acidic, 1 to 3 pH. The vaginal canal has secretions that are 4.4 pH. And then you also make antimicrobial molecules like antibodies, sebum. Okay, sebum is like oils that you produce on your face. Mucus. In your tears have lysozymes, and these lysozymes are enzymes that kill bacteria. So I guess that's one good thing about crying, is that you are killing the bacteria with the lysozymes. And then you have complement proteins. Complement proteins, along with antibodies, they actually break open, like they puncture microbes. So think of a bacteria, and then the complement system like breaks up their plasma membrane, okay? And then there's biological defenses, like you have phagocytes, natural killer cells that go around and they just like kill any microbe that may have, you know, that is on your skin or around you, okay? And then you have your own microbiome. The microbiome refers to the good bacteria that lives on you and in your body. They're there to, you know, take up space, and they actually help you. There is some bacteria that you have a mutualistic, symbiotic relationship. Remember the ones that help break down lactose, right, in your digestive tract? Okay, and they're there and they're taking up space. And when they take up space, there's no space for the bad bacteria to grow, okay? And in this PowerPoint, I have this other YouTube video that is good to watch, okay? Okay, you know, why are white blood cells called phagocytes or macrophages? Like, think about the word phago, macrophages. Like, the word phago and phage, you should think of phagocytosis. Do you remember what phagocytosis is? It's taking in solid particles. It's bringing it into the cell, okay? So this is a picture of a drawing of a macrophage, which is an immune cell that is innate, and they just go around and they look for microbes. So the cell membrane surrounds the microbe. The microbes get stuck in a vesicle. They get engulfed and they get stuck in a vesicle, or a vacuole in this case. Then the lysosomes within the macrophage are going to fuse with the vacuole. The lysosome contains digestive enzymes that are going to digest and break apart the microbes, okay? And then the microbial debris is released by exocytosis, okay? So this is how macrophages, phagocytes get rid of bacteria, okay? They're called macrophages, one more time, because we're talking about it's something big is coming in, okay? And I mean, bacteria are small, but it's not a food. It's not like a glucose molecule. It's a bigger cell, okay? And there's so many different kinds of phagocytes, or there's many different kinds, okay? Like there's the macrophage, neutrophil, free macrophage, like going through your bloodstream, looking out for invaders, the eosinophils, the monocytes. So there's a lot of different phagocytes, and they pretty much do the same thing. It's just they have different names based on the location in the body and what they specifically respond to. All right, the inflammatory response is innate. Okay, this is what you're born with. It's part of the innate response. Let's say you get pricked by a needle, or you get a paper cut. And when you get a paper cut or pricked by a needle, the needle, I mean, if it's not sterile, or a piece of paper has a lot of bacteria, and you're basically injecting into your skin bacteria, which are foreign invaders, okay? So when tissue is injured, there's going to be the cells around there are going to release a chemical signal, okay? And actually, it doesn't say it on here, but the cells that we're talking about that send these signals they're called mast cells, okay? These mast cells release chemicals that causes the inflammatory response, okay? And these chemical signals, an example of one is histamine, okay? Some of you that have allergies, you might take an antihistamine, because an allergic reaction, I should say. Allergic reaction is part of the inflammatory response, okay? Okay, and then what happens when those mast cells release the histamines? It increases blood flow to the area of injury, and when there's increase of blood flow, swelling occurs because blood is flowing to the area, and then the blood vessels that carry the blood to the area, they become leaky, and all of these phagocytes or macrophages, they'll like leak out into the area, and then like vacuum cleaners, they will swallow up all the bacteria, okay? Okay, and so that was the inflammatory response, okay, which is part of the innate immunity. And innate is a rapid response, okay, and these are all different kinds of cells and molecules that are part of the innate rapid response. And then there's the other part, the adaptive immunity, which is what you acquire. Acquired immunity or adaptive immunity, that means the same thing. It's a slower response, okay, and that has to do with B cells and T cells. There are some T cells that are around that are kind of generalists. They will attack foreign invaders, okay, but most of the T cells that we're talking about in this PowerPoint are like helper T cells and cytotoxic T cells, which are acquired. They are specific to a foreign invader. And B cells, they're so important because they're the ones that make antibodies, okay? So acquired immunity or adaptive immunity, okay, is what you build up in your lifetime, okay? So acquired immunity is what you build up in your lifetime, and there's two kinds of acquired immunity. There's active immunity. This is what when you're exposed to a pathogen, this is what you naturally produce. This is your immune response. And then there's passive immunity, where antibodies from another person, like a mother, when a mother is breastfeeding her child, the milk actually contains antibodies that will help the baby. The baby doesn't make the antibodies, but the mom does, and it'll help the baby not get sick against a pathogen. So there's two types of responses that is acquired or adaptive. Remember I said there's the humoral response. And when you think of the humoral response, you should think of the B cells. There's the cell-mediated response, and you should think of the T cells, okay? Cell-mediated, T cells. Humoral, B cells. And there's two kinds of T cells. There's the helper T cells, and then there's the cytotoxic T cells. And there's actually a few type of B cells as well. There's the memory B cells, and then there's the plasma cells that produce antibodies. We'll talk about antibodies in just a few minutes, okay? You know, B cells and T cells, they're also called lymphocytes. Okay, they're called lymphocytes. Both made in the bone marrow. So like in your thighs, your thigh bones are really big. Okay, your thigh bones are big. That's the bone marrow where your lymphocytes are made. Okay. I don't know if this is true, but I've heard that this is true. Okay, I should have verified before even bringing it up. But you know, like when you feel sick, you might have like aches, like they say muscle aches or whatever, but sometimes you might feel aches in your bone. I don't know if this is true, but I've heard that it's your body like trying to pump out lymphocytes. I don't know if that's true or not, but when you're sick, the bone marrow is where the B and the T cells are made. Okay, so but then once they're made in the bone marrow, they'll be immature, and then they go to a like the T sites, the immature T cells, not okay, lymphocyte. It's an immature T cell. They go to the thymus, which is, you know, right over here, and the thymus is where they mature, and then they become a T cell. Okay, so they become a mature T cell. Let me move my face over here, maybe. Okay, whereas the lymphocyte immature B cell, it's going to remain in the bone, and that's where it's going to become a mature B cell. Okay, that's why it's called B versus T. B for bone, that's where it grows up. That's where it's born and grows up. And then the T cell, it's born in the bone, but then it matures in a different city, in the thymus gland. Okay, so B for bone, T for thymus. So the humoral versus cell-mediated, okay? Humoral, again, when you hear the word humoral, you should think B cells and the antibodies they produce, whereas the cell-mediated, you should think of T cells like the helper T cell, that's what the TH stands for, helper T cell, versus the cytotoxic T cell, okay? So the humoral response is the B cell activation. You know, when a B cell encounters an antigen, so what is an antigen? An antigen is the foreign thing, okay? It's like the virus, it's a bacteria, it's a pathogen. Or in the case of somebody with allergies, it's pollen or whatever you're allergic to, that's the antigen. Antigen binding to the naive B cell receptor. So this is a young B cell, and this is the antigen. It binds to the receptor. And then what's going to happen is that B cell is now activated. It's going to start to divide. It's called clonal selection, because you have lots of different B cells. You have B cell 1, you have B cell 2, you have B cell 3, 4, 5, 6. Okay, lots of B cells, but they need to be activated. Once they bind to the antigen, they're activated, and then they make copies. They make copies of themselves. They're like building up an army. They're building up an army against that specific antigen. Okay, and then the naive B cells, they can turn into a plasma cell or a memory B cell. So some of those B cells become a memory B cell, which you keep in your body for a prolonged period of time, just in case you encounter the same antigen again. Or it becomes a plasma cell. And these plasma cells have a lot of rough ER. Do you guys see this? That is rough ER, because rough ER is where the ribosomes are attached. That's where the proteins will be made. And those proteins that I'm talking about are antibodies. These are antibodies, which are Y-shaped proteins that attach to the antigen. You can see that here. They're like bound to the antigen, and they will cause the antigen to become immobilized. And the antigen can no longer cause an infection because they are immobilized by the antibody. Okay, So what are antibodies? I said they're Y-shaped proteins. So this is an antibody, antibody, antibody that bind to antigen's epitope. So these are 4 different antigens, foreign substances, okay? And epitopes are where they bind, where the antibody binds, okay? So the antigens are like the foreign thing, and then the epitope is where the antibody binds. So what do antibodies do? They immobilize the pathogens, okay? So if this is a virus, antibodies will surround it, bind to the epitopes. In this case, those are the spike proteins, okay? And now that virus with those spike proteins, it can't bind to the receptor protein of the host cell, and it won't be able to enter the host cell. Okay, remember how viruses get in? They have to be welcomed in by the spike protein attaching to the receptor protein, and it's welcomed into the host cell. Okay, that's how viruses have evolved. Okay, whereas if they're surrounded by antibody, there's no way it can get in. Okay, once the virus gets surrounded by antibodies, immobilized by antibodies, then here comes a phagocyte. A phagocyte, a white blood cell, is going to take it in and get rid of that virus. So these are some of the things that antibodies do. What I just described to you guys was opsonization. Opsonization is when the antibodies surround the microbe or the virus, and then the phagocyte comes around and swallows it up. There's also something called agglutination. Agglutination is where the antibodies chain the bacteria together, so the bacteria cannot get away and cause an infection. Okay, it's sort of like, you know, when criminals get arrested and they get handcuffed together. So you can just imagine the bacteria getting handcuffed together. Yeah, so those are just some examples of what antibodies do. And there's 5 classes of antibodies. Antibodies are also called immunoglobulins. Immuno because it's part of your immune system. Globin because globin refers to proteins. Okay. IgA is found in mucus, saliva, tears, breast milk, protects against pathogens, and it's made up of several antibodies. Okay, like 2 antibodies together. There's IgD, which is a single antibody, IgE, IgG, and then IgM. Okay. You know, B and T cells can bind to antigens too. Okay, that says 2, T-O-O. So this is a B cell, and B cells, remember, they're the ones that make antibodies, but B cells can have, it looks like antibodies on the surface of their cell membrane that bind to specific antigens, okay? And these receptor proteins, you have the C region, C for constant, meaning that this will always have the same amino acid sequence. Remember, these are protein. Same amino acid sequence. Whereas the V regions, the variable regions, have differing amino acid sequences. And because it's different amino acid sequences, you're going to have different structure, right? 3-dimensional structure, which is going to be specific to the antigen that it binds to. Like, it's like a lock and key, right? The antigen and then the variable regions, they like fit together, okay? And then the T cell, like the cytotoxic T cell and the helper T cell, they both have 2 chains of proteins, the constant region, which always stays the same, and that's the variable, that's the antigen binding site, that's going to be different. Okay, so here you have like a variety of B cells that are specific to antigens, and once this B cell binds to an antigen, then it starts to divide and clone itself. Some of the B cells will become memory clone cells, like stick around your body for a long time and remember to keep an eye out for these specific antigens. And then some of them become plasma cells, which pump out antibodies, right? And that's what this is. These are rough ER and Golgi. Rough ER and Golgi, where the antibodies are going to be made and modified and then secreted out of the cell. Okay? Whereas cell-mediated immunity refers to the cytotoxic T cell or the helper T cell. Let's talk about the cytotoxic T cell first. Think of it this way, cytotoxic. Like when I think of cytotoxic, I think, oh, that sounds bad, right? Yes, it is bad for the cell that's infected, because what a cytotoxic T cell does to an infected cell is that it kills the infected cell. So bad news for the infected cell, okay? I can't speak. A cytotoxic T cell, it has a surface protein called CD8, and that CD8 protein helps that cytotoxic T cell grab on, latch on, and hold on to the targeted cell, infected cell, okay? So this is a cytotoxic T cell. This is a CD8, okay? And that CD8 is going to bind to a receptor protein that's found on the surface of the target cell that's infected. And that surface protein, that receptor protein, is called class 1 MHC molecule, okay? So all of your cells have class 1 MHC complex, okay? Your cells make the MHC class 1. And the cytotoxic T cells, the CD8, can bind right to it, okay? And so what's happening here is that this infected cell, it's infected. It has an antigen inside of it now, like let's say a virus inside of it. Well, a piece of the virus is going to end up on the outside. It's going to end up on the outside of the cell, because what happens is that when it becomes infected, the MHC molecule is synthesized, it's made, and then a piece of the antigen attaches to it, and together they go outside the cell. It's almost, think of it this way, it's the body cell that's infected showing a piece of the antigen to your immune system, like, hey, I have an infection, I've been infected with this specific virus, here's a piece of the virus that I'm infected with, now go kill and look out for these guys, okay? So it's like sending a message to the outside world, like, hey, I'm infected. And the cytotoxic T cell binds using its CD8 and its T cell receptor protein. It binds to the MHC class 1 molecule, and then it's going to release enzymes called granzymes and proteins called perforin and granzymes that are eventually going to poke holes into the cell membrane of the infected cell, make holes or pores, and then apoptosis. That cell's going to die, okay? And in the process of dying, you know, it's also killed off the antigen as well, okay? Whereas the helper T cell does not end up killing the infected cell, but in fact, the helper T cell works along with macrophages, okay? So the helper T cell helps macrophages. A macrophage will take in a bacteria or a virus, and then that macrophage will take a piece of the antigen on their class 2 MHC molecule, okay? MHC protein. So all of your body cells have class 1 MHC, whereas macrophages, like there's a macrophage called the dendritic cell, they have class 2 MHC molecules. Well, the helper T cell has a protein called CD4, which latches on to the class 2 MHC molecule along with the T cell receptor, and it communicates. Remember, this is all about communication. The antigen on the the antigen on the class 2 MHC molecule is going to communicate with the helper T cell. That helper T cell latches onto the MHC class 2 molecule via the CD4, okay? That helper T cell, as a result of, you know, communicating with that dendritic or macrophage cell, is going to release cytokines, and cytokines are chemicals that are going to activate cytotoxic T cells, activate B cells, okay? And those B cells that are activated is going to cause the humoral response, okay? Those B cells are going to be activated and make copies of themselves. They're going to clone themselves. Some of them will become plasma cells. Some of them will become memory B cells. Remember we just talked about that? Whereas the cytotoxic T cells that are activated, they're going to attack infected cells, okay? They're not going to attack the macrophage because the macrophage is not infected. Remember, macrophage is what destroys the pathogen, but they can go look for any body cell that is infected, okay? So the summary of humoral versus cell-mediated. Humoral has to do with B cells. Once the B cell is activated, what we call clonal selection happens, where they make clones of themselves specific to that antigen. Some of them become memory cells, some of them become plasma cells that pump out antibodies, okay? Whereas the cell-mediated, cytotoxic T cell, when it encounters a infected cell, it's going to destroy the infected cell. It makes memory T cells, and it makes active T cells that go out and kill body cells that are infected. Whereas the helper T cell, remember what it does? Right here, the helper T cell is going to release cytokines that are going to activate cytotoxic T cells and B cells. So that's what's going on here. The helper T cell activates helper T cells, and then and you can see the arrow going this way, cytotoxic T cells, and it activates the B cell. Maybe that's why they're called helper T cells, because they activate all the different kinds of cells, okay? So when you are exposed to a pathogen for a second time, we call that the secondary immune response. So one of the, I mean, I'm sure some of you guys have heard like, oh, you never get the same cold twice. Why do you not get the second cold? Why do you not get sick from the same cold virus a second time? Is because of the secondary immune response. The first time you are exposed to that cold virus, antigen A, you know, your body slowly responds. By 7 days, you start to make antibodies against it, and then you start to feel better, okay? Because now you're getting rid of that virus. But then the second time you get sick, you get exposed to it again, your body reacts a lot faster and stronger. It makes a lot of antibodies. It makes a lot of the B cells, the T cells. It's just the response. It's faster and longer and more strong, and that's why they say you don't even feel the sickness, because your body fights it very quickly. Okay. Okay. We're almost, I think, more than halfway done. Okay, now we're going to talk about animal development. All right, let me just press pause here just for a sec. It does turn out that I am recording, which is good. Okay, so anyways, now we're going to talk about animal development. Here's a beautiful picture of us when we were at 5 weeks old, okay? How did a single cell develop into this intricately detailed embryo? Well, it all starts at fertilization. And when the sperm fertilizes the egg, then the cell starts to divide. And that cell division is called cleavage, because it's like the cell is cleaving. It's like cutting in half. It's becoming two cells. And then gastrulation, that refers to a process where cells, you have thousands of cells or hundreds of cells, and eventually you're going to form a gut, okay? Gastrulation is basically the formation of a gut. And then organogenesis, and that is exactly what it sounds like, is when you start to make organs. Like one of the first organs that's made is actually your brain. And then morphogenesis, remember, that's taking form. So you actually start to see pattern formation. Pattern formation is like where the arms grow, where the legs grow. It's like morphogenesis. You're taking form, you're taking shape, okay? And then, like I said, this is all about cell communication, right? The development of an organism begins with communication between the sperm and the egg. And you can see here that once the sperm contacts the egg, it releases these enzymes that I guess eat away or digest that, or yeah, digest the egg's jelly coat, and then the plasma membrane of the sperm touches receptor proteins on the egg. If they can make a connection, if like a lock and key, they make a connection, then the sperm is welcomed in. The nucleus of the sperm is welcomed in, and that's what's going on here. That's the nucleus. Oops, let me go back. That's the nucleus of the sperm. And it is welcomed in. And then remember, sperm nuclei have half the number of chromosomes as the egg nuclei do. And so they are haploid. Remember haploid? They're both haploid. And when fertilization takes place, when the egg and the sperm fuse, you get a diploid organism, 2N, a zygote. Once sperm nuclei enters the egg is activated with the release of calcium ions. And do you guys remember where are calcium ions stored? In the smooth ER, the endoplasmic reticulum. And look at this. Doesn't this look kind of familiar? Okay, this is a signal transduction pathway. Okay, All these arrows mean there's like this activation. I remember we did talk about this, the PIP2, remember that? The tyrosine kinase pathway, the tyrosine kinase pathway causes PIP2 to split into DAG and IP3. IP3 binds to the endoplasmic reticulum. Calcium ions are released. You guys, that calcium ions, when it's released, it's so important because the calcium ions that are released by the smoothie R activates the egg. The egg starts to make mRNA and proteins that are going to cause the cell to divide, and cleavage is going to happen, okay? And so, again, it's fertilization, cleavage, which is cutting, which means cell division, and then you end up with a ball of cells, and that ball of cell is called a blastula. It's a hollow ball of cells. And then gastrulation, and you can see a little bit, okay, like a gut is forming, a tube, mouth to anus, okay, gastrulation. And then organogenesis, that's the formation of organs. And then morphogenesis, in this case, is called metamorphosis, okay? Organogenesis is the formation of organs. I like this because this is a gastrula. Okay, remember I talked about gastrulation? Well, a gastrula forms, and a gastrula has 3 layers of tissue in humans, okay? In humans, the outer layer of cells is called the ectoderm. The ectoderm eventually becomes your skin, and then over here you can barely see it, but the ectoderm becomes the neurons in your brain and your central nervous system. Whereas the mesoderm, like the middle layer of tissue, becomes your heart, cardiac muscle cells, skeletal muscle cells, cells that make up the tubes of your kidneys, red blood cells, smooth muscles in the gut. And then the endoderm, the internal tissue, it's going to become the lung cells, the thyroid gland, like most of the glands, okay? And then digestive cells. All right, morphogenesis. I mentioned morphogenesis is taking form. That's pattern formation, the spatial arrangement of the body. Like, how does your arms know to grow out? How do your legs know to grow out? There's positional information. There's positional information in the form of molecules that tell cells, you know, what they're going to become. For example, how does a chicken wing form? Okay, you might be thinking, why are we talking about chicken wings? Well, they did a lot of experiments on chicken wings because chicks can easily be, you know, grown and manipulated in the laboratory. And you don't necessarily have to have a shell either. Like, you can chip away at a shell and you can actually see the chicken growing, okay? But they've done a lot of experiments on chickens, and they have found that the limb bud, okay, that limb bud, this thing, which is going to become the wing, those cells that are part of the limb bud, they get positional information from molecules, okay? And when I say positional information, it tells the cells in the limb bud, where is the anterior? Anterior is like, where's the front? So this is the front. Anterior means front. Where's the posterior? That's the back side, okay? Where's the proximal? Okay, proximal means like the center of the body. Where's the distal? Okay, and you can't see that because my face is covering it, but proximal and distal are like opposite to each other. Proximal is like the core center of the body, whereas distal is out there, the fingertips when I put my arms out, okay? So the positional information are in the form of molecules that are released by several types of tissue in the limb bud. For example, the ZPA. ZPA is part of the limb bud that's on the posterior end of the limb bud, okay? So when I say limb bud, I hope you know what I'm talking about, okay? It's what's going to become the wing. It's like this bump. It's a bump on the embryo, okay? And that's going to become the wing. It's going to look like this eventually with the bones and everything. But on the posterior part of the limb bud, there's tissue called ZPA, okay? ZPA. And ZPA produces a protein, and this protein is called sonic hedgehog, okay? Named after, as you guys know, the video game character, okay? I think the guy that discovered ZPA and this protein, or morphogen, he named it Sonic Hedgehog because that was his favorite video game at the time. Okay, so ZPA produces a molecule called Sonic Hedgehog. Sonic Hedgehog is a morphogen. A morphogen is a molecule that helps with pattern formation or morphogenesis, and it tells the limb bud where is the posterior of the wing or the hand is going to end up. Okay, so like if you put your hand out, and we have a ZPA too. We have ZPAs and we do produce Sonic Hedgehog as well. Okay, when you put your hand out, where your pinky is, okay, like where your pinky is, that's the posterior part of your hand, okay? And in our limb bud, okay, the ZPA was on the posterior part and it told us how to grow our hands, okay? So this is a limb bud of a human embryo, and the ZPA is located on part A, and ZPA produces hedgehog, the morphogen. The pinky finger area has the highest concentration of hedgehog, while the thumb has the lowest concentration. Okay, so they did this experiment not on human embryos, I should say. They did an experiment on chickens where they didn't inject actually. They took a ZPA from one chicken and they added the ZPA to another chicken wing limb bud, and they ended up having a chicken embryo with 2 ZPAs. And when you have 2 ZPAs, you're going to end up with two chicken wings from one. I can't really draw it, okay? But it's a chicken wing. Let me go back to here. So what happened was it ended up having like another chicken wing tip over here. So there were like 2 wing tips because as it was growing and developing, the ZPA, because it had 2 ZPAs, it thought it had 2 posterior ends, okay? And so you ended up having like a double wingtip on a wing, okay? So there's Sonic. Okay, we're going to talk about the nervous system. So, you know, the nervous system is made up of neurons, okay? And neurons, my goodness, their job is to communicate with other neurons, okay? You have, like, each neuron has hundreds, if not thousands of dendrites, okay? Dendrites are where they communicate with other neurons. And so one neuron is communicating with thousands of other neurons. How do they communicate with each other? Well, this is the basic neuron structure. You have, I mean, neurons are cells, so and cells have a nucleus. So you have a cell body with the nucleus where the DNA is found. And then you have the dendrites, the tips, where they're communicating, receiving. So the dendrites is where they receive. They receive ligands. They receive the signal. So they receive a signal at the dendrites, okay? And then you have the axon. An axon is a part of the cell that's super long. And that axon is usually covered by a fatty cell called Schwann cells. And that makes it the myelin sheath. It's like basically insulation. It's insulation. And then an electrical impulse is going to move down the axon. And then the electrical impulse is going to leave. Or not leave. It's going to reach the axon terminals. And the axon terminals is where the ligands, the neurotransmitter proteins, are released. Okay? So a synapse is where 2 neurons are almost touching. They're not touching. So this is called paracrine signaling. Paracrine signaling, where they're almost touching, where the neuron sending a message releases neurotransmitters to the receiving neuron. Okay, so these are 2 neurons. The synapse is where they are almost touching. So this is the axon terminal. This is the terminal where you have vesicles filled with neurotransmitters or ligands or signal proteins. Once an electrical impulse reaches the axon terminal, those vesicles release neurotransmitters. Okay, there's so much involved with this section, but I don't think it's going to be, I'm going to skip through some of the stuff. Okay, so here's a nice picture of a synapse. This is the cell, what we call the presynaptic cell, meaning like before the synapse, okay, the space between the 2 neurons. It's releasing the neurotransmitters, the ligands, which are received by these receptor proteins on the postsynaptic cell. Okay. So how does information flow through neurons? They are received by the dendrites. The electrical impulse is going to go through the cell body, and then the electrical impulse is going to move down the axon, and then it's going to release neurotransmitters. It's going to pass on the signal to another cell, like another neuron or another effector cell. When I say effector cell, like it could be a muscle cell, maybe it causes muscle contraction. Okay. So, I'm just like debating whether or not I should go over this. I think I will super quickly, okay? So why are all cells in your body? Okay, all cells, if you were to take a voltmeter, and a voltmeter, it measures voltage, electrical voltage. If you were to take a electrical voltmeter, sorry, and measure the voltage inside of your cells, they would all be negatively charged. Negative 70 millivolts is the electrical charge inside of all of your cells, including neurons. And it's maintained this way because of the sodium-potassium pump. Remember the sodium-potassium pump? It's going to pump 3 sodium positive ions out of the cell, and it's going to pump 2 potassium ions in. And then also on the inside, there's a lot of like chlorine. Oops, I can't spell. There's a lot of like chlorine ions, okay, Cl ions. There's a lot of like negatively charged proteins. But the main way that it's maintained is that you have a lot of positive charges on going outside, and then you have fewer positive charges going inside. It's like a 3-to-2 ratio. There's more positives going out than there's coming in, okay? So we call this an electrochemical gradient, because you guys remember when you talked about concentration gradient. A gradient is like a difference in concentration across a membrane. Like when you have a cell membrane, you might have more of a solute outside than in. That's a concentration gradient. With this one, we call it electrochemical because we're talking about electrical charge, negative versus positive on the outside. And it is chemical too because we're basing this on ions, like sodium ions, potassium ions, chlorine ions. So based on ions, chemicals, we're forming a gradient, an electrical chemical gradient. Okay, so all cells have a negative 70 millivolt charge at rest. I should say at rest. A neuron at rest has a resting potential of minus 70. Okay, so this is what the cell membrane, the plasma membrane of neurons and all of our cells look like. There's all these different ion channel proteins. Okay, this is the sodium potassium pump. It's pumping 3 sodiums out, 2 potassiums in. And then, you know, some of these ion channels, like they open up, they close. Okay, you have potassium ion channels. You have sodium ion channels. Okay, in addition to the sodium potassium pump. So, you know, the sodium potassium pump, it pumps 3 sodiums out, 2 potassiums in. But then you also have individual sodium channels and potassium channels. Okay, once they open up, they let the sodiums in, and then once they open up, they let the potassium, once the potassium channels open up, they let the potassiums out. Okay, so what's happening here is that when we say, oh, there's an electrical charge, you know how I said that earlier? Oh, there's an electrical charge that moves down the axon. What do I mean by that? Well, electricity is the movement of like ions, right? And in this case, we're talking about when there's an electrical charge here, the sodium ion channels open up. And remember, sodium is like, by the sodium potassium pump, it's pumping a lot of sodiums out, right? There's a lot of sodium ions outside. But when a sodium ion channel opens up, sodium will rush in, and now the inside of that cell right here becomes positive. It becomes positively charged. Remember, the resting potential is negative 70, but when the sodium ion channels open up, it becomes positive because sodium ions are positive, okay? So there's a current flow due to opening of sodium ion channels. When it becomes positive on this section of the axon, it activates the sodium ion channels in this section to open up. So you can see that's happening here, okay? Those sodium ion channels open up, which causes the next section of sodium ions to open up. And that's what's happening here. So when the sodium ion channels open up, it opens up in this, they open up in that direction. It's like a domino effect, I guess you can say, a domino effect where they all open up sequentially, okay? But here's another thing, is when right after the sodium ions come in, then they close, the ion channels close, but then the potassium, the K pluses, their channels open up. And remember, potassiums are found inside, K plus are found inside the cell because that's what the sodium potassium pump does. So when their channels open up, they go outside. And you guys think about it. If the potassium ions, which are positively charged, start to leave the axon, what type of charge is going to be formed inside the cell? You have positive charges leaving, then it's going to become negative again, okay? So an electrical impulse is when the sodium ion channels open up and the axon becomes positive, but then it goes back to being negative when the potassium ion channels open up. They leave, positive potassium ions exit the neuron, and they go back to being negatively charged, okay? It's kind of confusing. So that's what's happened. I think I should have probably described that when I went over this. So sodium ion channels open up, sodium rushes in, it becomes positively charged, which causes the next section of the membrane, sodium ion channels to open up, and they rush in. That's what's happening here. But once the sodium ion channels are opening up here, then the sodium ion channels over here, they close up, but then it's now the potassium ion channel to open. It's their turn to open up, and they're going to exit the cell. And because potassium ions are positively charged, it's going to cause the cell to become negative again. Okay? So they turn positive, and then they go back to negative. Okay? So I feel like I don't need to go over this, but this is like measuring the voltage inside of a cell during what we call action potential, okay? And action potential is this electrical charge moving down the axon. Okay, and so what happens? Okay, we're talking about communication. Let's get back to communication. When the electrical impulse or action potential gets and arrives to the axon terminal, where the axon ends, that's why you call it an axon terminal, the depolarization, which means that it's becoming positive, okay, positively charged. When the axon terminal becomes depolarized or the positive charge reaches the axon terminal, it's going to cause these calcium ion channels to open up, and calcium is going to come in. Okay, calcium ions are going to come in, and when they come in, they attach themselves to these vesicles. And when they attach themselves to the vesicles, it causes the vesicles to move to the plasma membrane for exocytosis. They're going to release the neurotransmitters. The neurotransmitters will bind. See this right here? They bind to the receptor proteins, and these are what do we call them? Gated ion channels. So the neurotransmitters are the ligands. They're the proteins. They bind to these ion channels, causing the ion channels to open up. And you guys, when they open up, positive charges like sodium, calcium, they come in. That's an action potential that occurs in the next postsynaptic cell. This is the postsynaptic cell. This is the synapse right over here. And then this is a postsynaptic cell, which is the cell that receives the ligand, the message, okay? So once the cell becomes positive here, then that's an action potential that's going to move through that cell, okay? All right, and there's lots of different types of sensory neurons, like there's the photoreceptors, which we talked about early on. There's chemoreceptors that, so like, with chemoreceptors, it's chemicals that bind to receptor proteins. And when they bind to receptor proteins, it causes like the ion channels to open up, and there's an action potential. It stimulates that neuron, like taste. The chemicals in food. The chemicals in food are on your tongue. They bind to receptor proteins. Ion channels open up, and then it's stimulated, and you can taste the food. That's what we really mean by tasting the food. When we taste the food, it's really because the chemical in the food is binding to that receptor protein and causing an action potential, and we recognize that as a specific taste. Oh my God, that sounds so bizarre, doesn't it? Smell. Smell is the same way. Particles in the air go up into your nose. They bind to receptor proteins. Ion channels open up. Action potential tells your brain, whoa, I smell such and such, okay? And olfactory receptors, that's what we call the receptor proteins. I'm sorry, not the receptor the receptor cells, okay, that detect odor, smell. And then there's also mechanoreceptors, like touch, pressure, pain. You know, like when sometimes you're like just kind of resting and then you feel something tickling your skin. Maybe it's a bug or a spider crawling up your arm. You can feel that because of the mechanoreceptors. Hearing. Hearing is like, you know, the eardrum is vibrating, which causes action potentials in your ear to send a message to your brain, the auditory part of your brain, etc. Thermoreceptors detect temperature. Okay. All right. The last thing we're going to talk about, oh my goodness, we're almost done, is cell communication and the cell cycle. Remember the cell cycle? Okay. Remember it's how a cell, when a cell's born after mitosis and cytokinesis, it grows in the G1 phase and it gets ready for a cell, it gets ready to synthesize DNA, replicate DNA in the S phase, and then the G2 phase is getting ready for mitosis, and then the M phase is mitosis and then cytokinesis, right? Well, there's several checkpoints in the cell cycle, and these checkpoints are very important because they tell the cell if it's ready for the next stage of the cell cycle. You know, the G1 or the S checkpoint is the start or the restriction point. Like, is the cell big enough? Has it grown big enough and it's ready for synthesis of DNA? Remember, if the G1 or the S checkpoint, if there's no go-ahead signal, then it's going to enter the G0 phase. Remember the G0 phase? G0 phase is when the cell is never going to divide, right? So checkpoints regulate progression through the cycle. And what are these checkpoints dependent on? They're dependent on cyclins and cyclin-dependent kinases. So the CDKs and the cyclin, when they come together, they're the go-ahead signal for the cell to move on to the next phase. And also, besides cyclins and CDKs, you can have external growth factors that can stimulate cell division. Okay? Here's an example of a CDK cyclin complex, MPF, or mitosis promoting factor. The mitosis promoting factor is part of the G2 checkpoint. If the cyclin and CDK come together, they make the MPF promoting factor promoting mitosis. It's the go-ahead signal for the cell to go into mitosis, okay? And you know, the amount of cyclin in the cell, it goes up and down, or inside the body it goes up and down. Inside the tissue it goes up and down. The cyclin, when it's available, it will bind to the CDK and cause cell division, okay? But if there's no cyclin around, then it's not going to go through the cell division, but it's going to help regulate the cell cycle, okay? You know, the cell cycle is so important with all of these checkpoints because you know, mutations can happen, and sometimes mutations in like tumor suppressor genes, or like, for example, P53, which we'll talk about in a second. You know, if there's mutations, usually apoptosis will occur. The cell will send a signal that it needs, that there's, you know, errors in it, and then it will commit cell death, program cell death. But cancer is essentially a failure of cell division control. So instead of the cell dying by apoptosis, it continues to divide and grow unrestrained, uncontrolled. And why is this? It's because the checkpoint stops are lost, okay? The gene for P53, it creates a protein that it's really key, that plays a key role in the restriction point of the G1S phase. And what it normally does is that it halts cell division. It causes the cell to go into G0 if it detects damaged DNA. But if there's a mutation, there's no halting of the cell cycle. The cell cycle will continue and it can lead to cancer. Okay, that's what that says there. It says it's unable to halt the cell cycle and it leads to cell division out of control, which is cancer. And I am done with this PowerPoint. Oh my goodness. So I hope that wasn't too bad. I hope that you guys were able to learn a lot there. So anyways, I will see you guys, talk to you guys later, and have a good day. Bye.